As you can hear, I am under the weather, and I have this very wonderful voice right now that you're going to have to listen to for the next 30 minutes or so. But in any case, I'm really delighted to be here with you and talk more about honeybees and the factors that affect their health in agroecosystems. So maybe some of you watched the first part of the seminar last week, and we're going to continue with this talk, especially when we dwell more into the factors that affect honeybee health. So last week we mentioned what ecosystem services are and the economic impact of honeybee pollination and pollination services that are done by honeybees in not just the U.S. economy, but also the global economy. So today we're going to talk about factors that influence honeybee health, and that includes also the problems associated with agrochemicals like neonicotinoid systemic insecticides. However, I want to put a word of caution. A lot of the research that has been done with neonicotinoid insecticides is undergoing, and a lot of the studies have been done only with topical applications on individual bees, meaning that the amounts of chemicals that the bees are being exposed to are only on a per capita, per bee basis on their feed, or topical application with some sort of acetone-based diluent, and then putting bees in cages to see their lethality or the sublethal effects of these, or the LD50s or the lethal doses of these chemicals on the bees. So a lot of the field relevant studies that we want to see on nicotinoid insecticides and their effect on colony growth are just underway. So the jury's still out, and we're really excited to, or anxious to see results at the colony-wide level. So you have to be a little patient with what I'm going to show, because it is kind of just the beginning of the story, what we've done so far. So what are some of those factors that influence honeybee health? Our poor honeybees are not only the most influential pollinator of a third of the food we consume, just like I talked about last week, but they are in charge, while they're doing all of these pollination services, they are also being kind of attacked from every front, and including pathogens, pests, and disorders. Poor nutrition or starvation, the effect of pesticides, and the effects of climate and environmental changes. So what are some of the pathogens, pests, and disorders that are afflicting our honeybees? First, I want to go over what we consider pathogens. So those are infective agents that grow within the bees. Then we have parasites, which are the infective agents that feed upon the bees, so ectoparasites, they're outside. We have pests, so those are animals that attack the honeybee colonies for food or other resources. And we have other factors that are not due to parasite, pests, or pathogens, but are still problematic for beekeepers. And these pest pathogens and other factors can be transmitted horizontally between bees when they are sharing food or when they're feeding the young, or vertically from the queen being transmitted through the egg to the new young. So what are the important brood pathogens? And of course, I could spend maybe 3 webinars to go over each one of these factors. So this is just kind of one of the very short summaries of all the things that are ailing the bees on top of our neonicotinoids and other agrochemicals. So in the brood, that means the babies, the larvae, and the pupae, we have all of these things that are detrimental to honeybee health. We have bacterial diseases like the infamous American foul brood, for which the regulatory inspection, apiary inspection services were created to abate the AFB in the United States in the early 1900s. Because of all the big work that was done by the apiary inspection services across the U.S., AFB is now quite contained because it's very contagious, and the only way that we can get rid of it is by eliminating colonies altogether. Then we have the European foul brood, also a bacterial disease, and these are pictures of how you can test for these diseases. You have something known as the rope test. You have to go and look at the very slimy brood and see if what comes out is kind of a gooey, long, mucus kind of thread that comes from a toothpick to the dead and diseased brood. We have European foul brood. That's another bacterial disease. Then we have viruses of the brood, like sac brood. As the name indicates, the brood gets kind of wrapped in this sac, and the brood gets killed inside, changes coloration, and it's also quite severe. And we have fungal diseases like chalk brood, as the name indicates. The larvae and pupae turn into these mummies that look like pieces of chalk, and they're also quite damaging. So those are brood pathogens. Then we have very important adult parasites. Arguably the most damaging one is the parasitic mite Varroa destructor, which got introduced, as I mentioned last week, into the United States in the 1980s. And it switched hosts from an Asian honeybee to the European honeybee, which we have here, and has been quite damaging because the bees did not have the immunocompetence toolkit to combat the varroa mite. So they're ectoparasites because they live on the outside. You can see here a drone, a male bee, with a varroa mite on his thorax. It's sucking the hemolymph or blood of the bee while transmitting more than a dozen viruses, and also creating lesions through when they're sucking through the cuticle of the bee that can cause infections at the incision side. But varroa mites love brood, so they attack larvae and pupae, and that's their preferred mode of action because they can just stay inside the capped cell for several days. So this is arguably the most damaging parasite that we have in the U.S. currently, and what is causing most of our bee issues, even more so than agrochemicals, anyone in my field would argue. Then we have tracheal mites, which are these tiny microscopic mites that live inside the breathing tubes of bees, these tracheae, and they also will feed on the hemolymph of the bees, but because they're so large compared to the size of the tracheae, they don't allow for proper respiration and gas exchange inside the bees, and you can see these damaged tracheae with all of these mites inside. And then one of the most prevailing current problems is the Microsporidia Nosema apis and Nosema cerati. There are two species. They're difficult to identify amongst each other microscopically. You have to do it with genetic testing, but this is what they look like under the microscope. You can do some counts of Nosema per bee. They cause distended and darkened ventriculus, which is the midgut of the bees where most digestion takes place. And these Microsporidia, they come in spores. They get ingested as spores by the bee horizontally through the oral fecal route, and then the spores germinate inside the bees. They throw out a filament that goes attaches to epithelial cells, and that is where the cells reproduce and create more and more spores per bee. They cause a very symptomatic hive streaking. This is a picture of a hive with a very severe problem with nosema, causing bees to causing diarrhea because they cause digestive issues in the bees. And so bees are very clean generally. A healthy hive will have foragers that will void themselves or go and defecate outside of the colony so that they can avoid contamination of all the things inside the hive. But when they are having issues with nosema, they can't contain it because they get dysentery, and so this shrinking pattern is obvious, and it shows that the bees are not digesting their food properly, so they're malnourished and they collapse. Then we have other pests that are not pathogens or parasites, but are still problematic. We have the wax moth, which is a moth that will the larvae will feed on, as the name indicates, the wax in the colonies, and so they will make their cocoons, and they will basically burrow throughout the comb and making it basically useless, especially damaging all the honey crop. Then we have in the southern states a very big issue with the small hive beetle. For both of these issues, strong colonies do not have big issues with these two pests. Wax moths and small hive beetles are more, they are not the causal agents of colony collapse or weakness. It's actually a secondary symptom of already weakened colonies. So colonies that already have other issues with pathogens will succumb to the the small hive beetle or the wax moth, and so the small hive beetle will lay hundreds of eggs in the colony, and you can see the little larvae will just burrow through all the pollen and honey stores and eat it. So when strong, a colony just gets rid of the adults or keeps them secluded, but when weak, these adults reproduce very heavily and damage all the honey and pollen crop in the colony. And then you have other problems with insects and mammals, like wasps that try to go into hives and usurp or rob colonies. Or this is what is the colony collapse disorder in some areas of the country, where this is a collapsed colony because a bear attacked the colony trying to get all the honey. So in a lot of areas, you might need to put wire fences, electrical wire fences around your apiary to avoid the disruption of colonies by raccoons and bears and other animals that like to eat the food inside the hive. So this is now as a joke, we say this is the true colony collapse. I'm not going to go into colony collapse disorder right now because you could spend also a whole hour just talking about it, but I just want to mention that it's now being called other things by other people, like multiple or multi-caused syndrome, colony death syndrome or something, because we basically don't know what causes colonies to collapse. It's a combination of all of these things that I'm talking about today. We also have other factors that are not due to pathogens, pests, or other animals, but they're still a big problem. In areas where there's very harsh winters, we have issues with chilled brood. So the brood always needs to stay, the brood is the babies, and babies need to be at a temperature that's around 35 degrees Celsius, and so the bees that take care of the brood will shiver and produce body heat that will keep the brood warm even in the cold winter. So it's a way for thermoregulation. But when it's too cold and the colony is too weak, then there's not enough workers to keep the brood warm, and so the brood gets chilled and dies. Short brood patterns are those that are caused by poor quality queens that are quite inbred genetically. Excuse me. And so you start seeing poor laying pattern. We have also an issue with laying workers. You can see these cells have several eggs per cell, and that's a very key indication that the colony is probably queenless, and these workers will start laying unfertilized eggs that turn into drones, the males, not the workers that perform the of the tasks, the work tasks. Drone-laying queens, so when the queens are not properly mated, they can start laying unfertilized eggs because they don't have sperm to fertilize eggs, and so this dome-capped brood here is drone brood, and that's really not desired if you want to have good honey production, because the drones will not be collecting any food. Then we have issues with poisoning, and that's kind of what we're going to be talking about neonicotinoids and other insecticides, and then issues with poor queen quality that leads to the workers replacing their mother queen in a situation like this one where you have drone-laying queens. The workers will decide that the queen is not performing her duties as the provider or creator of workers, and so they replace her with a new queen, and thus it disrupts the brood cycle because it takes a couple of weeks for the new queen to come out, mate, and start laying eggs, and it also changes completely the genetic makeup of the colony because that queen will be mating with a drone or a set of drones that are completely different from the ones that her mother had previously mated with. So those are the pathogens and pests and other factors. Now we're going to talk about nutrition and or lack thereof that can affect honeybee health. There is a critical role of pollen diversity on honeybee health, and this is particularly of interest to people in the green industry. You, just as experts in this field, would absolutely agree that polycultures are more diverse and therefore more nutritious to pollinators than monocultures, whereby the pollinators of the crops only have access to one specific type of crop for an extended period of time. So it's been shown that dietary protein diversity, that is diversity in polyfloral pollen, has a direct impact on the immunocompetence of honeybees. So it's recent studies have shown that polyfloral diets, that is the diet that comes from bees being able to collect pollen from a variety of sources with different amino acid contents, different amounts of and quality of proteins, have a better in-hive antiseptic protection, so allows bees to have this social immunity to combat diseases, and they have higher amounts of amino acids that are required for tissue development and growth, for example, the production of fat bodies and also the development of hypopharyngeal glands, which produce a lot of the brood food that is necessary to raise healthy brood. So if you can imagine, instead of this purple lavender field, if you can see thousands and thousands of acres of almonds in California right now, basically in February, the bees only have access to one type of pollen for the duration of the pollination bout, like 3-4 weeks. And all they're eating is pollen from almonds. And so they're only receiving the amino acids that are present in almond pollen. And that is not something that anyone, including the bees, would want to have. Just a very boring and maybe a diet that doesn't have all the essential amino acids for proper tissue development and growth. So then we can talk about pesticides, and this is, I guess, what you guys are most interested in, and so we'll go into this into detail. So you might already have knowledge of what systemic pesticides are, but those are the ones that neonicotinoids are in. They're basically seed-treated. The seeds are treated with these insecticides, and when they germinate, the insecticide is going to be inside the system, inside the plant, and all the parts of the plant are rendered poisonous. So the stem has the insecticide inside it, the leaves have it, and it helps them combat things like mites, leaf miners, aphids, wilt, But one thing that was not really thought about very much was that these systemic insecticides can also be inside pollen and nectar of flowers. So these systemic insecticides are chemicals that are absorbed by the plants through the roots and render the parts poisonous. Sorry. And some have a highly specific affinity to the nicotinic acetylcholine receptors of insects. And these insects and arthropods like mites, leaf miners, and aphids are they have the nicotinic acetylcholine receptors, but so do bees. So it's really difficult to have these insecticides work and operate on trying to combat these insect pests without also alerting or affecting, at least sublethally, honeybees. So it's been shown that systemic pesticides, when applied in high doses, are extremely toxic to bees. One of the studies that was done 3 years ago that kind of brought about the notion of the effects of chemicals, of agrochemicals, or the amount of agrochemicals in honeybee colonies was this very highly cited paper by Mullen et al. Out of Penn State of high levels of miticides and agrochemicals in North American apiaries. So the authors collected wax, pollen, and honey and honeybee samples from over 250 apiaries in the United States. And this was published in PLOS One, so I recommend that you download it for free and you can get all the information about what's in hives and in bees. To cut the story short, all the paper shows they did liquid chromatography from all of these samples to determine the levels of these chemicals in each type of sample collected. So, for example, here, so there's several tables on that paper. This table happens to be the one that collected samples in the agrochemicals in wax, pollen, and bee samples. So, a few things come to mind. The top two chemicals that were found in wax, pollen, and bee samples, not surprisingly, were the pyrethroid fulvalinate and the organophosphate coumaphos, which are the two most widely used beekeeper-applied miticides to control the varroa mite that I just talked about. So it's been, they were heavily used. Fulvalinate was heavily used in the 1990s. Coumaphos was heavily used in the 2000s. But because the mites have become resistant to these two products, we've kind of run out of we've stopped using them. But because the wax is highly lipophilic, that means it acts like a sponge for all of these chemicals, and because we don't change wax very often as beekeepers, all of these chemicals get trapped in the wax year after year. So the two most commonly used chemicals, fulvalinate and coumaphos, were found in 98% of the samples of wax, about 80% of the pollen samples, about 60 to 80% in bee samples. So that's not gratuitous. Then we have other things like chlorpyrifos, another organophosphate, chlorothalonil, which is a fungicide, amitraz, which is the currently most widely used miticide to control the varroa mite, which is a formicidal mite. But then you can go down the list right here, and it tells you what kind of chemical it is. The table continues. So here we have, I don't know, 3-4 dozen chemicals, and then it goes down even further down, and you start seeing that imidacloprid, the neonicotinoid, systemic neonicotinoid, it's not in the top chemicals found in wax or nectar or pollen. It's not up there. It's kind of toward the end, and it was only found in 1% of wax samples and 3% of pollen samples, and in no bee samples on the bees. Then we have thiacloprid, which is the one that was found first, and it was found in 5% of pollen samples and 2% of wax samples, but that's not really high compared to the other types. So I just want to bring this up for you because neonicotinoids, even though they are pervasive, in some cases, they are not the most widely found chemicals in wax and pollen samples across the United States. So what are some of the neonics, or some of the studies that have been done with neonics in the United States and abroad? So one of the first studies that was done was with imidacloprid. Imidacloprid was actually banned in the 2000s in France because of citizen complaints and beekeeper lobbying efforts, because they noticed that sunflower seeds treated with imidacloprid were claimed to be the culprit of colony collapses in France, because they claimed that the sunflower pollen and little nectar was contaminated heavily with imidacloprid, even though those levels have not been widely shown scientifically. How did people start looking at these studies of the effects of these chemicals on bees themselves and their health? Well, the first type of experiments that are done are those that are done, as I said at the beginning, topically on bees. So you take a bee and you apply the pesticide on their body, and they see through translocation, because bees groom themselves, they are going to start grooming their body with their mouth parts, and so some of those molecules of the chemical gets translocated into their body and then can affect metabolism functions. So the first type of studies that are done with these kinds of chemicals are typically these PER, or proboscis extension reflex experiments. So to do this, you harness a bee so that you only expose her head. So bees smell through their antennae, and they have their mouth part here. It's called the proboscis. It's the tongue that comes out and collects nectar from flowers or a food reward. So these PER experiments are done to show, to test the levels or the learning and memory capability of bees, depending on what kind of diet they are fed. So PER extension reflex, proboscis extension reflex experiments are kind of simply done by providing first a food sample, sugar syrup, for example, that has a specific type of odor. And so you are trying several times to condition the bees to associate this specific smell, for example, lemongrass, to the fact that they are receiving a food reward. And so they, at the beginning, you train them several times to providing a food sample with an odor, the lemongrass, for example. Then after several times, several attempts, the bee will only need to receive a little puff of lemongrass odor for them to start extending their proboscis, because they are associating the smell with a food reward. And so people can test learning and memory, meaning how are they likely to remember whether this smell is associated with a food reward, and how many times it takes for them to associate the food with the smell with the food reward. And so that's what we call conditioned responses. And so in this experiment, when bees were fed sucrose with imidacloprid, so compared to controls which only had the sugar syrup, there was significant lower learning memory and memory in honeybees. So here we have these 3 different trials. We have the control, so by the second conditioner experiment, about 90% of the bees had learned to associate the smell of the food with the food reward. There was no significant difference between the controls and very small amounts of imidacloprid. But when fed high levels of imidacloprid, so that is by 3 orders of magnitude or more, the bees took a long time to get to associate the smell with a food reward. So that means that the bees are not learning fast enough compared to controls how they are likely to get a food reward just by giving them the food sample. But this type of experiment was the first one done, but it also showed that the exposure to imidacloprid has to be at very high levels, and these levels are not field realistic. They're much, much higher by orders of magnitude than what that they would get exposed to if they were in the field. Another very recent study, though, shows that neonicotinoid pesticides reduce bumblebee colony growth and queen production. So this is one of the first ones that we really want to see out there showing us true results. So this study took bees, but again, this is a problem. They fed bees individually with a pipette, and they fed them either clean sugar syrup or sugar syrup that was contaminated with either low, so we have here clean sugar syrup, and then low in the solid line and high in the large dashed line, levels of imidacloprid in the sugar syrup. So the first 2 weeks, these bees were in an incubator, and they were fed these levels of food in an incubator. And then they were taken to the field, so they were taken to put inside colonies, and then they weighed the colonies here as far as how heavy the colonies were. These were done with bumblebees, and bumblebee colonies are very small. So they were able to take all of the workers or most of the workers in a colony, set them, and then put them back into their colonies. So it's easier to do this with bumblebees, because if you take 60 bees, that might be 80% of the worker population in the colony. You can't really do that with honeybees unless you took 1,000 bees and fed 1,000 bees this kind of food and put them into a colony that is 1,000 times, that only has 1,000 or a couple thousand bees. So that's why the study was done with bumblebees, because it's kind of strength in numbers. So lower colony weight was shown over time in colonies whose workers were exposed to low or high doses of imidacloprid. Then, sorry, I'm going the wrong way. In the same study, they also looked at queen production, and they showed that in colonies that were exposed to either low or high levels of imidacloprid, there was a highly significantly lower number of queens that were produced in colonies. That is incredible, and that is something that needs to be done with honeybees, but it's already showing incredible repercussions in the bumblebees, which are also some of the most important pollinators that we have in North America. Another study that was done with the neonicotinoid thiacloprid showed that it says bees under stress, sublethal doses of a neonicotinoid pesticide and pathogens interact to elevate honeybee mortality across their life cycle. So in this study, the authors tested the interactions of field realistic doses of thiacloprid and 2 microbial pathogens, Nosema cerana and black queen cell virus, individually and in combination, on larval development and also on adult mortality. So black queen cell virus is a virus that happens during larval development, and as the name indicates, the queen dies because of this virus, and you can see the queen inside turns dark brown to black, hence the name. And then, as I showed you earlier, Nosema cerana, which is one of the 2 Nosema species that affect honeybees. So they wanted to see the interaction between 2 common problem pathogens and thiacloprid. And so in adults, here, I don't know if you've seen in the green industry what is known as the hazard ratio. So it's just a number. It doesn't have any units, and it just kind of allows you to compare how hazardous chemical is, all else being equal in industry or in a test. So here we have colonies that were controlled, so colonies that did not have exposure to either Nosema or black queen cell virus or thiacloprid. And these are the ones that are individually exposed. And so there's really no negative, the hazard ratio is not positive. But as soon as you put Nosema cerani and thiacloprid, or Nosema black queen cell virus and thiacloprid, you have an elevated hazard ratio, meaning that thiacloprid is acting as a synergistic element to increase the potentially damaging effects of Nosema cerana and black queen cell virus. In this study, they also looked at, so this one here is on adult survival, adult mortality, and so it increased adult mortality when you had a combination of the thiacloprid and the two pathogens. Here they are showing the additive interaction between black queen cell virus and thiacloprid on larval mortality compared to controls, and the hazard ratio is very high when you have here thiacloprid and black queen cell virus together presented, and you test larval mortality. So this one shows that additive interaction between those two because likely the pesticide elevated those viral loads that are detrimental to the colony. So those are three examples of experiments that are being done to show the the effects of thiacloprid, imidacloprid, amongst others, which are the two most highly found pesticides, neonicotinoids on wax samples, pollen, and bees. And finally, I just briefly want to mention that honeybee health is also being affected by changes in climate and environment. So the climate might change the phenology of honeybee pollinated plants, causing early plant flowering or later plant flowering and nectar flow. So it kind of disrupts the natural patterns of foraging that you can see throughout the year in a specific area. And the climate can also change the geographic distribution of flower species that the bees depend on for food. Climate can cause excess drought or rain, which can reduce the nectar and pollen in quantity, like it happens in some areas in France. So if you have excessive drought, the flowers will stop producing nectar that is collected by bees normally in that area at that time of year. Or it can do the opposite. If there's too much rain, the rain can wash off the nectar that is being produced by the flowers, causing basically the same effect, no nectar for the bees, causing death by starvation. So as you can see, the equation is a very delicate one that has all of these players. And I would argue also that on top of all of this is management or poor management. And what we're doing as beekeepers and or farmers is heavily influencing the honeybee beekeeping industry. So what is making our bees sick are the way that we produce agricultural products might be making our bees sick, and in turn, sadly, poor pollination services by honeybees because of their weakened immune system or lower numbers are causing our food products to become of lesser quality or amounts, especially as we are increasing our population and our food demands. The bees will not meet the demands, the pollination demands for our food industry. So complete pollination produces fruits and vegetables that are symmetric and larger in size. We, especially in the first world, are very spoiled. We have beautiful, we like to see beautiful fruits and vegetables that we can consume, and we like them because they're symmetric and plump and juicy. But the lack of available pollinators to pollinate crops can lead to incomplete pollination, which produces fruits that are asymmetric, smaller, and not as nutritious. We don't want it to go into a situation that is seen in other countries where the lack of honeybees because of contamination by agrochemicals and other air contamination has led to some areas in Japan to people having to pollinate their crops by hand. So we don't want to really go into that. But The good news is that, as you saw, some of those studies with neonicotinoids, plus all the studies that we in the bee research industry are doing, are helping increase the health of honeybees and raising awareness to people like you folks in the green industry about better management techniques so to avoid inadvertent exposure of bees to agrochemicals. For example, we are encouraging the production of nectar corridors. This is an example of a pollinator seed banks that shows what is available in that area that should be provided to native pollinators. People like Marla Spivak at the University of Minnesota, she has developed a selective genetic line of hygienic bees that have a behavior that naturally makes them be more hygienic toward varroa, and so they will get rid of varroa without the need for beekeeper-applied pesticides. Organic farming helps reduce the amount of chemicals used in crops that are pollinated by bees, so that helps. Honeybee health research, like the one that we perform in our bee, we look at the effect of agrochemicals on the reproductive health of honeybees. And citizen concerns, like the Sierra Club, for example, in the United States has been trying to ban imidacloprid and neonics because they want to help save, bees and bee habitats. We now have the presidential mandate for pollinator protection programs. We have the EPA mandates on new labeling on chemicals that can be detrimental to bees when exposed to them. So anyway, we're basically, the news are kind of grim, but they're also hopeful in that we're doing a lot of things to try to combat all of these ailments that are coming from left and right and top to bottom to the poor bees. So in conclusion, many factors, diseases, disorders, nutrition, climate, the environment, and pesticide use influence honeybee health. Is not just one thing that we can look at in an isolated manner. Some lethal levels of agrochemicals, including systemic insecticides, can accumulate in wax and or on bees to cause detrimental effects on bee health and longevity. And novel research, beekeeping, and farming practices will help improve the quality and quantity of our honeybee populations, especially in an expanding global market. So I want to close, you might have seen this before, I want to close by saying that our diet is heavily influenced by the variety that is provided or allowed to us by the pollination of a lot of crops by bees and other native pollinators. So our breakfast table would look kind of like this, or looks generally like this. With honeybee pollination, we have fruits and vegetables that are pollinated by bees, jams. You can see all the nuts here, especially almonds and other fruits, cranberries. Then we have juice, fruit juice, and we have even milk that is produced by cows, but the cows are feeding on the alfalfa that is pollinated by honeybees. So you can see how the chain can be kind of divided by several degrees of separation. And this is what the food will look like. So we would still be eating food, just granola, the wind pollinated crops, regular coffee, although coffee is also pollinated by bees, so maybe we should take the coffee out, eggs and wheat toast, but no diversity in our food. So anyway, I want to leave you with that. Here's a picture of our website, so you're welcome to go and look at our website. This is our interim president, Dr. Hussey, with our Aggie Honey. Please visit our Facebook page, which is more updated. It has a lot of, I just posted this right now, for example, which might be of interest to you. It just came out of Dartmouth University, showing that secondary metabolites in floral nectar like nicotine or the terpenoids such as thymol, which is surprisingly or not surprisingly is the product that is being used as one of the alternative controls for varroa mites, form part of the bee's natural medicine cabinet to combat disease. And so it's a good idea to plant plants that have these secondary metabolites in them so that bees can collect them to control disease inside their colonies. And with that, I leave you. Thank you so much for your time, and I will be happy to take any questions.
Dr. Rangel, we have, I don't know whether you can see that in the chat box. There's one question is, you know, when the bees were transported to multiple states for pollination, does the travel cause stress to the hives?
Absolutely, yes. I have a colleague out of Michigan State who has been trying to get funding, although he has been unsuccessful so far, to mimic the stress caused by traveling thousands of miles for pollination trips. So he did a preliminary study. He was able to take these test colonies and take their levels of stress genes before putting them in a truck for transportation to pollination, and then during and then after the trip. And he saw lower amounts of these, so upregulation, what we call the increase in the expression of genes that are related with stress. So now what he's doing, because he wasn't able to get more funding for this preliminary study, what he's doing is he's mimicking the shaking of bees in a truck. So he like inventively put bees in a cage inside a shaker that will shake bees for like 24 hours for 2 weeks, and measuring their levels of gene expression based on this, and he has seen some very interesting results of increased expression of genes that are associated with stress. So yes, absolutely, that is one of the, not only because of the movement and because they're being caged for so long, but also because when they get released, they are basically like this mega colony where it's a lot of drifting going on, and the potential for a lot of disease transmission, horizontal disease transmission between colonies.